we get a number of different characteristics of muscle out of that arrangement of how muscle contracts. Okay? So muscle tension or pressure or force can only develop where actin and myosin are interacting, right? That means if I take a muscle, think of that sarcomere picture. If I pull the sarcomere apart, at some point the actin and myosin filaments aren't anywhere near each other anymore, right? So that muscle can't contract. Similarly, if I squish that muscle together, the actin and myosin filaments are now all bunched up in the middle and they can't interact properly either. So that means that we have this relationship between muscle length. Yes, ma'am.
I'm so sorry. So with like spasticity after a stroke, is that then constant release of calcium in the areas of where you have spasticity, or is that?
I mean, technically, yes, it is, but it's because of neural issues. So upper motor versus lower motor neurons. So in a stroke, you've taken away the control, so the lower motor neurons are on all the time. So they're telling that muscle to contract, and the cerebral cortex isn't in control anymore. So it's, yes, there's lots of calcium around, but it's because of the neural system. Because like you can stop that spasticity by severing the nerve, right? And then that goes away. Does that answer your question? Okay. All right, so we have this relationship between muscle length and muscle tension. Length, okay, amount of tension developed. We see that the maximum is sort of in the middle, right? B and C, where there's lots of room for interaction between the thin filaments and the thick filaments. Between B and C, the sarcomere is working like it's supposed to. All right, well, so let's take an example where we've stretched the muscle too much. That's D. So when a muscle gets very long, its tension falls to 0. Why? Because we've literally pulled the thin filaments away from the thick filaments. So they can't interact, they can't contract. Myosin can't reach actin, right? So it can't contract. In A, we have the case of a squeezed, squished muscle, right? Where the actin and myosin filaments are so closely bound together that they can still interact, but they can't develop very much tension. All right, so do we see A and D in the body, in the natural human being? We don't, right? Because of how muscles are arranged in pairs, and because of the anatomy, the body works to keep muscles in the right length zone. So the only time you see this fall off in D and A is experimentally, where you've taken a muscle out of an animal, stretched it or compressed it, and measured it. But the reason that I share it with you is it sort of proves that the shape of the sarcomere is in fact what causes contraction. Because if we disrupt the shape, either in the too long or too short direction, then the system doesn't work as well. So it's a bit of proof that the cross-bridge cycle is really how the muscle contracts. Another way we describe this is as the sliding filament theory of muscle, where you have two filaments that are sliding next to each other. Okay, so in anatomically, whole muscles are sort of prevented from this problem because of the anatomy. All right, so however, there is still this effect that when a muscle is too short or too long, it's weaker. All right, so here in this graph, we have, now we're away from the sarcomere and we're at an actual muscle, right? So here's the tension of the muscle. Here's the length of the muscle. So you can see that strength is greatest, right? Just a little bit longer than normal. Now, why is that? Because when a muscle is stretched just a little bit longer, it's much more like C than B or A. In other words, it optimizes where the thick and thin filaments are, and therefore allows much greater strength. Now, the place that this plays in skeletal muscle, this doesn't really play much of a role because muscles work in pairs, right? So when one's contracting, one's relaxing. But when we get to the heart muscle, this notion of length and tension becomes really important because it underlies what we call the Frank-Starling mechanism of cardiac output control. In other words, if the heart is a little stretched before it contracts much harder and much more completely than it does if it's a little too short or a little too empty at the beginning. And that's directly because of this relationship between the sarcomeres and the filaments.